Welcome everybody. My name's Pip. I work in the communications team at MDI BL. I want to thank you all for being with us for this amazing Science Cafe. So tonight's guest, we're incredibly lucky to have her. We wish we could have had her in person. Tonight's speaker is Dr. Navas-Acien. Last time she was at MDI BL was 6 years ago, so it's taken a while to get her back, but here she is. And she just told us before we all logged on that she's got some exciting research coming up, so we'll have to definitely invite her back in the near future to share that all with us. She is a professor of environmental health at the Colombia School of Public Health, and her scientific research focuses on the role of environmental exposure and chronic health conditions, so how those two things connect and play out. So today she'll be talking specifically about arsenic exposure and the link to cardiovascular disease. This is particularly pertinent here in Maine because we have a high dependence on well water for household supply, but we also, especially in coastal Maine, sit over an arsenic belt, which means we have a higher prevalence of higher levels of arsenic in our water if we rely on well water. So we have had a long-standing and impressive history here at MDI BL of connecting with toxins that affect the environment. And at the moment we have one of our own faculty, Dr. Jane Disney, running a program called All About Arsenic, which is a program for school students, and we've got 12 schools in Maine and 6 in New Hampshire. And what they basically do as part of this program is they collect community well water and it goes off to be tested, and they basically learn to use their scientific literacy that they're learning to advocate for their community and raise awareness of the trouble with arsenic. But that's kind of aside because we've got Dr. Navas-Acien here to tell us more about her research. So welcome, Anna. Nice to have you here.
Thank you, Pete, so much for the invitation, the opportunity to be here. Thank you also, Jerry Bowers, for this opportunity. I am very sad not to be in Maine, at Mount Desert Island with you today. I have amazing memories from 6 years ago, so I hope to be back there again. The only advantage about us being remote is that I can see some familiar names of real arsenic experts out there who have joined this talk, and other people who might also be interested in arsenic and for whom it would have been hard to go to Maine today. So I suppose there are always plus and minuses. So as Pip mentioned, my training and my focus is in epidemiology, and I've done a lot of work on arsenic and specifically on cardiovascular disease. So I am going to be focusing particularly in that area of research, and we are going to go into a journey going around across time. We know a lot about arsenic for a long time. When we did first find out that arsenic could be bad for the cardiovascular system, I want to take you to those old studies. What's happening today? What do we know? How do we take that how do we take all this information, the epidemiological evidence, the experimental evidence, how do we take it? How do we inform the risk assessment? What can we do to protect our populations? So this is what I want to cover today. So with that introduction, I am going to share my screen and my slides. The first thing I want to highlight, when we think about cardiovascular disease, people generally think about lifestyles, your food, your diet. However, the WHO, the World Health Organization, is increasingly recognizing that a large portion of cardiovascular disease could be due to environmental risk factors. Right now, this is due to 31% has been estimated from air pollution, from secondhand smoke and chemicals, but only lead has been included. Arsenic has not yet been included. And this idea of that we have a discipline called environmental cardiology is a new idea. It's a 21st century idea. In the 20th century, lifestyles, diet and food were the major culprits believed to be important for cardiovascular disease. And this is really growing in the literature, this idea of an environmental cardiology. Regarding the metals, we have strong evidence for cardiovascular effects for arsenic, cadmium, and lead. And there have been systematic reviews in the literature published in very well-established clinical journals, such as the British Medical Journal, summarizing the growing number of evidence and of studies supporting that these metals are important for cardiovascular disease. I am going to focus today on arsenic, which is our element of interest, also because arsenic is the #1 toxicant of priority, as indicated by numerous agencies. And why is arsenic so important? First of all, because it's very toxic, as we are going to see, but also because arsenic is really widespread in the environment, particularly in water and also in food. And as Pip mentioned at the beginning, you have an arsenic belt in Maine. Arsenic is in the groundwater in many geological areas around the United States and around the country. There are lots of subgroups of the populations, especially in rural areas, especially in places which rely on groundwater as the main source of water, which can also happen in urban settings. When groundwater is important, arsenic could be there. But also arsenic is also in food, especially in the rice. And arsenic can get in the rice because of different reasons. In a major one is because rice really absorbs and requires a lot of water to grow. And arsenic is an extremely hydrophilic chemical. It really goes with the water, it travels with the water, and it accumulates in the rice grain. And there are also some other external factors, such as the past use of arsenical pesticides, manure that contains arsenical drugs when they were allowed in poultry production, some genetic types, forms of rice that are much better at accumulating the rice, or again, arsenic being naturally occurring in the soil and groundwater. But even on average conditions, whatever little arsenic is in the ground, it's going to travel in the water and it's going to end up in the rice. So that arsenic that you get in from the groundwater, from the rice, from some other foods that also contain arsenic, is going to end up very easily into our bodies. And arsenic, once we are exposed to it, especially the inorganic arsenic that we get from the water and the rice, is going to enter in the body, is going to be methylated through a quite complex methylation system that involves some enzymes that are becoming very well known in the literature. And through that methylation process, through the methylated compounds, especially the DMA, dimethylated arsenate, that compound is going to be very easily excreted. But all of the arsenic is more or less going to be excreted from the body, and it ends up in the urine, which is the main path for arsenic excretion. So in epidemiological studies, we often measure the sum of the inorganic arsenics, the monomethylated arsenic, and the dimethylated arsenic as the main source of, as the main biomarker for exposure to arsenic. However, arsenic exposure can be very complicated. For instance, if you eat lobster, as it's very common in Maine, lobster has extremely high levels of arsenobutane. Many other seafoods have arsenobutane and also other complex chemicals. All of them end up in the urine. So when you do epidemiological research, you really need to make sure that the species, the arsenic you are measuring, is reflecting the toxic inorganic arsenic that is in the water and in the rice to have a valid result. So I am, as I mentioned earlier, I am going to take you in a traveling in time, in a historical journey. And the first evidence on arsenic and cardiovascular disease related to a form of peripheral arterial disease in the lower extremities, in the arteries affecting the arteries of the lower extremities, that was first identified in Taiwan, but also among German Vietnams. And that type of disease was very severe. It resulted in an ischemia, necrosis. It was very painful, and it would result at the end in natural amputations of the toes and even of the full lower extremity in the more advanced cases. The interesting thing is that pathological, this disease was very similar in the description in the Taiwan cases and also in the German cases, which were exposed to those inorganic pesticides. Of course, this was all before internet and scientists being able to easily identify and connect areas of research. But there is an extensive literature actually going back to even the 30s and the 40s, documenting those findings. The second, this was extremely high levels of arsenic exposure, I want to clarify. The second important historical piece of evidence, and actually in Taiwan, this was then later on during the 80s and 90s, documented at in a very, what we would consider high quality epidemiological studies that were directed by C.J. Chen, an arsenic researcher who is now actually the vice president of Taiwan, and he's been credited with being in charge of the very successful COVID response. So I often, we can tell our students that if you do arsenic research, you can never know what can happen to you, and you might have a very successful career in other areas, as it happens with C.J. Chen. But in addition to Taiwan, and those studies from Taiwan, the second very historical evidence for arsenic exposure comes from Chile. And in Chile, there was a city called Antofagasta in a part of the country that is very arid, that suddenly in the late 50s they switched the water source and brought the water from a different river that happened to have levels of arsenic of 900 micrograms per liter. To give you a comparison, this is 100 times higher than our current MCL, 10 micrograms per liter in the United States and kind of globally. So extremely high level. This was a relatively large city, like around 300,000 people live there. And what they noticed in the 60s, 70s, is that there was an outbreak with people relatively young dying of cardiovascular disease. Even teenagers, even relatively young children were dying of cardiovascular disease. When they did the autopsies, they found thickening of the intima, which is the internal layer of the vascular of the arteries. And that was actually very similar to what the pathology that has been found in animal studies. And also they found this outbreak of cardiovascular disease, I am showing it here in men, for the years where the arsenic was extremely high in the water. And then the following years, it kind of went down, suggesting that if you remove the arsenic from the water, you may be able to actually control and mitigate the burden of the problem. This might not be true for other diseases, such as cancer, but it might be true for cardiovascular disease. So we have evidence from Taiwan, we have evidence from Chile, and I also want to bring you evidence from Bangladesh. And this is related to the work that the Colombia University Superfund Research Program has been doing for 20 years under the leadership of Dr. Joseph Graziano and Lex Van Geen. 2 years ago, I had the tremendous honor and challenge to become the director of the program. And this program has provided some of the most important evidence that we have today to understand the cardiovascular effects of arsenic. And the reason why this study was so important, and I am bringing you the main epidemiological study relating arsenic to cardiovascular disease, published in the British Medical Journal, is that for each participant in the study, 12,000 who were initially recruited in the year 2000, for each participant, they got a sample of their water, and they knew if the arsenic in the water was high in red or low in blue. And that distribution of arsenic in the water is geologically driven. It cannot be confounded as by lifestyle of the people or by knowledge of the people, because people didn't know. Arsenic is odorless, is tasteless. So this is the closest that you can get to a random experiment, especially at baseline when people didn't know their arsenic levels. Once people learned their arsenic levels and everybody was informed, then people started to switch. And switching was no longer at random because people with higher SES, more education, etc., started switching earlier. So if we want to study the change in arsenic exposure, that's going to be affected by confounding and other factors, but the baseline is not, because it can only be confounded by geological factors. It cannot be by human factors. And you can see there is a clear dose response with higher arsenic levels in the water being related with higher risk as measured with a hazard ratio, which is a measure of risk compared to the baseline, the reference, which is the group with the lowest levels. So clear dose response, clear excess risk. The good news in Bangladesh is that after 20 years of research, we've been able to reduce arsenic levels. And where before red was the dominant color, now we are very fortunate that most of the participants have actually low arsenic levels, less than 10 micrograms per liter. So these are wonderful news. And we really want to know what are the benefits for those changes. I think it's a question that many of you might have. If I have arsenic in my well, in my house, and I change it, am I going to see immediate benefits? Maybe I won't see any benefits, but maybe my children will see the benefits. How do we know? To do that, we need to do new research that study the change and what happens when arsenic exposure changes. So we've been in Chile, we've been in Taiwan, in the German vintners from a long time using arsenic pesticides, and we've been in Bangladesh. But what happens in the United States? Do we have any evidence that arsenic could be toxic at the lower levels of exposure that we have in the United States? So this has been an objective of my research, and that's what I want to share with you now. This was just a picture from Taiwan and from Bangladesh, showing also how different it is from the United States. And even though some of these toxic agents are likely to be generalizable, I think these are important questions to ask. Is the dose response, are the health effects similar or not across diverse populations? So in the United States, groundwater is the major source for inorganic arsenic exposure. And thanks to the work from the USGS, and this is work done primarily by Joe Ayotte and many others, and some of his collaborators as USGS, we now are able to know what is your probability of arsenic higher than 10 anywhere where you are in the country. And this is extremely useful for people who are using groundwater. And this is really based on domestic wells around the country, because anybody can have a sense of what is their probability, and then that might be very useful for deciding where to monitor or where not to monitor. So as it was mentioned, Maine is a place where definitively you want to monitor if you have arsenic if you have a well, a private well. But not only private wells are important. In this map, this is a recent work done by a recent PhD student, recently graduated, where she combined data from the EPA, the 6-year review data, affecting community water systems. And you can see that even in community water systems, we have counties around the country that are exposed to arsenic levels above the current standard. And even above 5 micrograms per liter, which is the standard now in New Hampshire and in New Jersey, and the standard in other countries. We have places above 1 micrograms per liter, which is the standard in the Netherlands. So are these counties at risk of cardiovascular disease? Are they at risk of cancer, of other health outcomes? But today, let's focus on cardiovascular disease. How do we know? And one of the best ways to answer that question is trying to do epidemiological research. So one of the studies that has been able to provide some answers about those potential risks is a Strong Heart study. Strong Heart is a study of American Indian communities across North and South Dakota, Arizona, and Oklahoma. As you can see in this figure below, it shows the urinary arsenic levels of random people selected from the study, from the STRONG-HEART study, over a 10-year period. And even though arsenic has a relatively short half-life, as I mentioned earlier, and it's excreted through the urine completely, because people are drinking water one day after another, after another, after another, over 10 years, the people who had high arsenic levels at the beginning still had arsenic levels high at the end. And people who had lower levels at the beginning tended to have lower levels at the end. But we saw most of the variability was across people. And that was true. There were differences across sites, with Arizona having the highest levels. So the STRONG-HEART study, again, had some random geologically driven exposures that are good as a reflection of a potential natural experiment. And we are able to ask, is arsenic in those communities being a risk factor for cardiovascular disease? The levels we have here in that study, in the STRONG-HEART study, are very generalizable to levels affecting many rural and semi-urban communities across the country. This is how an epidemiological study is done. There is a baseline visit where the participants are recruited, they are followed over time. The STRONG-HEART study, in addition, recruited family members in the year 2000. So now we have a very complex information, which might help us to do genetic research. So we've been able to connect the baseline urinary arsenic data with the health outcomes, cardiovascular outcomes happening over time. The study could not be done without team science, lots of different disciplines working together. I think that's what we know more when you are an epidemiologist, is that nobody is important in a way. Everybody's contributing a piece, and it's the collective effort what really makes the research possible. And the research could not be possible without a strong support and partnership with the communities. And this is a picture from one of our annual meetings. We meet regularly with our tribal communities. We work together, we decide together the research that is being done, and hopefully also be able to bring back. So the study, we found an association. Unfortunately, we found that even at these low levels of arsenic relevant for US populations, we found an increased risk, especially for mortality, also somehow for incidence, although more attenuated. I am happy to talk about that if there are questions about why this could be happening. And I mentioned the dose response. It's very important to look at the dose response. And in this figure, which are put together through flexible splines, we can try to look at the dose response. What's happening at the low levels? Let's imagine we think, yes, above 10 micrograms per liter, thinking of the standard, which is equivalent to microgram per gram of creatinine, adjusting for a urine dilution. Above those levels, there is a clear excess risk. But what's happening at the lower level? Unfortunately, the STRONG-HEART study didn't have enough statistical power to look at the dose response. And while it seemed to be linear for all cardiovascular disease, for coronary heart disease, which is the major form affecting the coronary arteries, when you have an MI or a heart attack, the dose, maybe there was a threshold. So we don't really know what's happening at these low levels. In epidemiology, consistency of the findings and replication is very important. And we've been able to replicate a very similar dose response in Colorado residents as part of a study in the San Luis Valley County. We also have evidence from animal experimental models. And this is a work published recently by Coren Mann, a toxicologist who has looked at atherosclerosis or similar kind of a model for atherosclerosis in an animal model, in a mouse model, and has been able to show a very similar dose response at very similar arsenic levels in the water as humans are exposed to. So we have strong epidemiological evidence and we have strong experimental evidence. Sometimes I wonder, what else do we need to say that arsenic is a cardiovascular risk factor, because the evidence is so compelling. And this is another recent study done in the Strong Heart study where we look not just at the clinical events, but we are looking at the imaging of the heart. How is the heart looking? What is the cardiac geometry of the heart? And how is it functioning? And we can see that the heart looks thicker. The heart is working much harder when arsenic is present. And these are relatively young adults where we looked into this. And so these young adults are working harder, their hearts are not functioning so well, and ultimately are developing heart disease clinical events. So there was an editorial that came together with this paper in the young adults of the Strong Heart study, highlighting how important arsenic could be for the heart. And this was published in a major clinical and cardiovascular journal. So we've looked into many cardiovascular outcomes in the Strong Heart study. I summarized the clinical outcomes here. I've summarized the cardiac geometry, but we have evidence with atherosclerosis, peripheral arterial disease, risk factors for cardiovascular disease such as diabetes. So arsenic is truly pluritoxic for the vascular system and also for many other organs and systems. Arsenic was one of the first carcinogens known for humans. So one of the questions is, how is arsenic doing its effect? What is happening? How is arsenic so toxic? It seems to be that the arsenic metabolism, the metabolic processes through which arsenic is methylated in the body, is particularly toxic and can explain some of the toxicity for arsenic, especially for some of the intermediate species. And actually, Korenman has shown that the methylation is quite important for some of those cardiovascular effects. And arsenic can induce general mechanisms such as oxidized stress and inflammation, and alter actually the electrophysiology of the heart, resulting in numerous effects for the vascular tissues and ultimately subclinical and clinical effects. And one of the mechanisms for explaining the number, I went too fast, for explaining the large number of effects for arsenic could be through epigenetics and the effects that arsenic has in gene expression. So this slide is borrowed from Anne Bozak, one of our PhD students at Colombia, who is now a postdoc at Mount Sinai. And she wanted to communicate to our tribal communities what is epigenetics and why it can be important for some of the health effects that we see related to arsenic. So she had this idea of thinking that epigenetics is like highlighting what's important in a textbook. Not all the cells are like chapters in our book, and not all the chapters you need to read the same information that it's in the book. So the highlighting, the methylation allows you to tell the cell what's important, what needs, which enzymes, which proteins need to be expressed. So that was really useful, and the tribes really appreciated that information. So we've been able to analyze DNA methylation using a very well-known array that has provided us with an extremely rich information for a relatively large sample size. So this is the exciting research that I was mentioning to Pip that we are doing. And we are just starting to disentangle some of our very exciting findings related to arsenic and DNA methylation. We have a paper coming out quite soon in EHP. And also we are looking into how epigenetics can help us identify who's at higher risk of cardiovascular disease. So we have some preliminary findings that epigenetic signals can actually help us improve substantially our risk prediction for cardiovascular outcomes. And we are also identifying some of the signals, the genes that might be potentially involved. And in this exciting research we are doing, we are connecting how arsenic exposure as methylation signals which are the genes that are turned on and off by arsenic are actually the ones that are related to cardiovascular disease. So this is quite exciting and novel for us to be able to work on this. But let's go back to our question about this low dose arsenic and what's happening at the low dose. The Strong Heart study was able to tell us quite a bit about the levels that are important for US populations, especially in rural areas exposed to arsenic through private wells. But the Strong Heart study was not good enough to telling us what's happening if you live in a city and your arsenic level in your water system, maybe like in LA, you live in LA and your arsenic levels in LA are around 300 micrograms per liter. And that's a pretty typical level for LA. Is that bad for us? Is that bad for the million of people who live in LA? What about people eating rice every day? You know, lots of people, because of preference, ethnic traditions, dietary risk for some diseases, like such as gluten intolerance. For many multiple reasons, there are people eating rice every day. What's happening at those levels? Do we have a risk for arsenic? So we try to answer that question, borrowing as much data as possible based on the Strong Heart study, but together with other cohorts that have been published out there. And this was work that was done together with Aaron Barchowski and Catherine Moon, who was a PhD which I mentioned earlier. And many other people, like Margaret Caragas, which many of you might know, and many other investigators. And when we put the evidence together, there was again inconsistencies at the low levels. Sometimes the effect seems to be linear. Sometimes the purple line is showing us the nonlinear. Sometimes there seems to be a threshold. The problem is that the confidence intervals for the nonlinear model are extremely wide. So we cannot really tell with the data, the epidemiological studies available today, we don't know what's happening at the low levels. We need new epidemiological studies at the low levels. So we were thinking hard, how can we do that in the most efficient way? Because this could be very difficult to do. So we thought that one opportunity was to partner with a study called the Multi-Ethnic Study of Atherosclerosis. This is a study being done in 6 cities around the country. The cities include LA, Chicago, Baltimore, Winston-Salem, North Carolina, New York, and St. Paul, Minnesota. So that gives us quite diversity in terms of arsenic exposure in the groundwater and in the community water systems, and also for different groups of people, because the study includes white, Black, Hispanic, and Chinese Americans. So if we look at arsenic in the urine of the participants, you see that there is extreme variability in what urinary arsenic levels are, even though everybody's pretty low. So even our Chinese Americans, participants who had the highest levels, are only, their geometric means are only around below 5 micrograms per liter. The lowest participants were the white people from New York, some of the lowest, and they have 1.79. So there is like a 2-fold difference, a little bit more than a 2-fold difference between those with the lowest exposure and those with the highest exposure. So is this variability at the low levels, is this a risk factor for cardiovascular disease? Maybe yes, maybe not. This data from a pilot study in work led by Miranda Jones, who is now an assistant professor at Johns Hopkins University. But we are now doing more analysis for all the participants in MESA. So our lab is very busy right now, although we had a shutdown, so we had to take a break for like 3 months now. We were just today back into the lab. And so we are measuring arsenic exposure in the 6,000 MESA participants. You might think this is a lot of work to measure arsenic species in 6,000 participants, but MESA has measured huge number of biomarkers, others that are even less relevant or others that are even more expensive than measuring arsenic. So this is just a tiny effort, given all the work that MESA has done. And this new work that is expanding We are doing it together with myself, with Tiffany Sanchez, there's a typo in her name, who is now an assistant professor also at Colombia University. So we now have data for 1,000 participants, and we are able to look at the preliminary data. And we actually have a meeting on Thursday with the group to go over those. And these are, again, very interesting findings. But, you know, because it's so preliminary, just had a chance to look at these numbers for the last week. And so MESA is probably going to tell us what's happening. Is arsenic a risk factor or not for cardiovascular disease at levels that are relevant for people living in urban cities and exposed to very low groundwater arsenic, or exposed to rice, primarily rice arsenic. I wanted to just, as I am finishing in the last few minutes, I just wanted to mention very briefly again that genetics are likely arsenic methylation is likely to be very important in explaining some of these health effects of arsenic. And I've mentioned about the methylation process earlier. And we have data supporting, especially for cardiovascular disease. Unfortunately, we don't have enough power to look at it. But for other outcomes that are continuous, such as glucose levels or insulin resistance, we've been able to look at some of these interactions, effect modification. And clearly, arsenic methyltransferase is very important in how arsenic is methylated in our population. And there is clear distinction in metabolism profiles in the urine, depending on your genetics. And there seems to be an interaction between your genetics and your arsenic and some of the health outcomes. So this is an area where we need pooling data. We need larger cohorts, many more cohorts, to be able to look at this gene-environment interaction. But what is very interesting is that in a study that has nothing to do with environmental health, some cardiovascular epidemiologists looking at genetics for ischemic heart disease, they found a bunch of genes that have nothing to do with the typical targets for ischemic heart disease treatments. And one of the genes that they found was arsenic 3-MT. So that gene was related with cardiovascular disease in a very big study that included the UK Biobank. Interesting. So I am going to finish, but before finishing, I want to talk about something that is really very dear to my heart and that I've really learned. I am very thankful for working with tribal communities and indigenous populations. They've made me think very carefully who owns the data, how do we share data, and in which terms, who decides how is the data shared, and who is profiting from research. Obviously, universities are profiting. I am profiting myself. I've been promoted to a full professor through some of the work I've showed you today. The students are benefiting. They're learning a lot, they're getting careers, they are becoming a faculty themselves. But what about the communities from which we get the data from, and who are so much and so important for the science? How are they benefiting? So one of the things I learned from them is, and I borrowed this picture because the same way that tribal communities are protecting our environment and are against things such as the Dakota pipeline, our communities are also resisting unrestricted data sharing. They have many concerns of how unrestricted data sharing could be harmful for them or even for all of us in the future if we don't understand how these data are used or misused. So the concept that research needs to, and indigenous communities need to benefit from science is very important. So we need to think, how are they benefiting? How can they benefit at the local level? How can they benefit at the regional level? How can we increase resources, plan prevention strategies? How do we contribute ultimately to policy and guidelines at the country and global level, which are ultimately might also protect communities? I think it's very important to think of it. This is, if we apply this to our current COVID pandemic, we can see it's exactly the same. There are implications at the local, regional, and more global levels. COVID is a very interesting example because it's such an acute short-term pandemic. While our arsenic pandemic is very extended in time, is chronic in time, but it's affecting numerous vulnerable communities, lots of population that are affected by the policies that we have in place, by the interventions that we have in place. So it's really important for us to think about how we are using those data. For instance, in South Dakota, the Mini-Wechoni, which means in Lakota, water is life, was a big investment that has contributed to safer drinking water at the community level. However, this could not reach everybody, unfortunately, because some people live too far away to be connected to the pipeline. And there are still going to be lots of homes, households, which will not be able to be connected to a community water system. And also, even if you are connected to a community water system, maybe your community water system is just mixing water to make it as cheap as possible. And your standard, your arsenic level just goes a little bit below the standard. But you are not reaching the final goal, that is to remove arsenic completely as much as possible from the water. So in order to help and do something with our communities in the Strong Heart study, with the families, the households that could not be connected to the water system, to the community water systems, we put into place what we call the Strong Heart water study for private wells. And this is a participatory intervention where we are installing filters for families, and we are actually going to be looking also at some of cardiovascular outcomes. And we are working with the tribes, and we are working with several community, and we are really taking an ecological model to make sure we have strategies that take into account the individual, but also the context in which families and communities live. And so I'm going to finish with this, and I hope there are lots of questions and comments. I'm really looking forward to that. I think from the evidence that I showed you today, arsenic exposure is widespread, affects large groups of the population at different levels. In rural areas in particular, we have lots of families and communities affected by arsenic in the groundwater. In urban areas, we still have people affected by low arsenic in the groundwater, which could be a problem. And especially we have a lot of rice eating all across the population. We don't know what those health effects are. But the evidence is clear that arsenic is a cardiovascular disease risk factor, and cardiovascular disease should be included in the arsenic risk assessment, which is still ongoing, and that really needs to be finalized in order to have additional protections for the populations. So there's still research that is needed, and that's exciting. You know, I hope I can come back again and show you some of the mechanistic and low-level data that we are generating. And finally, in addition to the policy and the research, the clinical guidelines and the environmental cardiology should be including arsenic and also other environmental exposures when thinking of preventing cardiovascular disease. So these are the American Heart Association and the American College of Cardiology primary prevention guidelines, and they call them A, B, C, C, D, D, E, and E stands for exercise. And my argument is that we need to add an additional E that stands for environment. We also need guidelines that include the environment as part of protecting people from developing cardiovascular disease. I want to thank the communities and the participants of the Strong Heart Study and MESA for making the research possible. I want to thank the funding from NHLBI, NIH, our Superfund research program. And of course, I want to thank lots of investigators and the amazing students and trainees who over many, many years, and this is not an updated slide, have moved the science forward. And with this, I am looking forward to answering some questions. So I'm going to stop sharing so that I can get back to see you, or maybe you can see me better like this. So thank you.
Thanks so much, Anna. That was, a beautiful, in-depth, while still being broad look at something that affects a lot more people than I thought it would. So we've had a couple of questions come in, so I will just ask them if you don't mind. First one from Sally says, boiling your water could increase levels to toxic levels. Is there any correlation with coffee, tea, or soup drinkers? And how do we reduce our exposure?
Very good. So that's a great question. As we know, boiling, is not going to solve your problem. And it might make it worse potentially. I have not seen data with coffee, tea, or soup specifically. We have data with drinks in general. And we found that even people in our study, we found that people having a lot of soft drinks like sodas had higher arsenic levels. And I was really surprised by that. And one of the hypotheses that I have is because of all the ice that people are adding with their sodas, and that could be potentially another reason. With coffee and tea, I don't know if we looked specifically at coffee and tea. So I am going to look into it. If we didn't find an association. I am going to confirm because we look into a very long list of food items, and we clearly found an association with urine, with rice, with these sodas, as I mentioned, and juice, like apple juice. All of that has been very well known. And we also found another intriguing association with liver meats, and not only with that, with spam and things like that. So we were a little bit surprised, or with organ meat, sorry, not with spam, with organ meat. And we were wondering if some of the animals are also locally grown, could be accumulating the arsenic in their liver, and that could be a reason. And but yeah, I look into that. That sounds like an interesting question. But what I was going to say is, despite diet only contributed to less than 4% of the variability in urinary arsenic in our study, with the limitations of measuring the diet, very limited. Imagine we could improve much better, but let's imagine it's 10%. Most of the variability in our population, the Strong Heart study, comes primarily from the water, not from the food.
Thank you. The next question is, are you aware of any research that has specifically focused on the under 18 age group?
That's a great question. In our study, we looked into young adults, and what we call young adults were actually from 14 years of age, so like end adolescence, and then through less than 40 years of age. So that is not only less than 18, but there are some studies with young children, not young, but adolescents and children. Studies in Mexico that I can remember, they also found similar effects than what we found with the imaging of the heart, and similar hypertrophy of the heart, like that those effects could be already happening in children. And the studies that I brought from Chile, that's where they found the problem, is that there were young children and adolescents dying of cardiovascular disease in that city in Chile. And that's what kind of was a red flag of why that was happening. So there are some studies, and I'd be happy to provide them if the person wants to ask me for an email later on. I put my email in the slides, and please share it with people. I'll be very happy to give the references for the studies from Mexico, the studies from Chile, and the different studies in young people.
Great, thanks for being so willing to share the research for those who really want to dive in deep. Another question that came from MDI BL's Jerry Bowers is, there's evidence that COVID-19 targets the vascular system. Does exposure to arsenic increase susceptibility to complications from COVID-19?
This is a very interesting question, because we know that arsenic can affect the immune system. And I didn't really talk too much about that, but there is a very strong literature on immune toxicity, on arsenic and immune toxicity. And we know that arsenic exposure can make the flu more severe. And there is some research ongoing about even if does arsenic exposure kind of impede the response to flu vaccination. And there were animal studies showing that arsenic can induce a more severe flu. So this could translate to COVID or some other viral infections. As far as I know, there is no data on that question. I know that some people have indicated that maybe the Navajo excess risk could be related to higher arsenic levels. The Navajo are very clearly one of the communities most affected by groundwater because of where they are located in rural arid areas where groundwater is very important. But I don't think there is solid data supporting that. I know that there is a study from Wuhan, China, which is looking into that. That's one of the advantages that I am an editor for a journal. So you see those papers coming in through your desk. So I cannot really say anything to you, but more probably there will be something coming out about that. And I think in Wuhan, they have arsenic exposure. So, and maybe some other, for sure, other metals also. So something to follow.
Yeah, I think COVID-19 is going to bring out a whole bunch of interesting new paths for people to follow with all the web of connections between everything. Bob asked, in your study in the tribal areas where you've provided water filters, are you filtering out just arsenic or other contaminants too?
That's an excellent question. So what we are using is an absorption media that currently only traps arsenic. So that's a big limitation because actually in our study, we also have uranium. And not everybody has uranium. The correlation between arsenic and uranium is not perfect. Actually, it seems to be a little bit together, but not together. But some people have both above the standard. What is currently, we are using that as the limit for installing the filters. So some participants are currently being excluded from our study because they have both uranium and arsenic. And the Indian Health Service is responding to those families with additional strategies. I think there is a real need for research, for new tools and methods to be able to deal with multiple metals, because it's likely that when you have, we found also tungsten, the cluster of arsenic, tungsten, and uranium seems to be really important in the Southwest. So how do we protect families from those together? I think it's very important.
So the last question that's come up is just said, it's from Sally, who asked the question about tea and coffee. Because some people may increase their exposure inadvertently by boiling to purify their water. Are there studies of water levels at different times of the year, because it might be the arsenic might be concentrated in dry periods?
Okay, so our study, in principle, each participant was recruited at a different time of the year. And that's a great question because I have not looked into that. So that's why I love, giving talks, because you get questions that make you think. And we could very easily look if there is differences in exposure by time of the year, independent of location and other factors. So we could look into that, and then we could see if the dry periods are more important or not. In principle, from the data that I've seen, the temporal variation is not too big, with some caveats and exceptions. But people are arguing that climate change and the increase in dryness might actually influence arsenic levels in the water. So this is definitely something to look into it. And I think, I think the issue with the coffee and the tea is that it's the very important issue, and the soups is the very important issue that people, when they use bottled water, they think I am protecting my family because I am using bottled water. But they generally tend to use the bottled water for drinking water. They don't necessarily use it, think of it for cooking. And that's why it's very important to think that if you need to protect yourself from arsenic in the water, boiling is not going to help. And any water that you use for cooking is going to be important. So the families need to have a strategy for protecting themselves from all the water used for drinking and cooking in the household. But it's not easy, always.
Wonderful. Well, Anna, thank you so much for being here tonight with us and taking the time to answer questions. Your research is fascinating. Can't wait till we can have you back to talk more about the things we've got in the pipeline, excuse the pun. Everyone who's joined us tonight, thank you for being here. Our Science Cafes are always a great chance to introduce you to some guest speakers and extend your knowledge of different areas of science, of which there are so many. Anyone who's new to us, thank you so much, and we hope you enjoyed your first Science Cafe. It's a series we run as part of our mission to create higher levels of science engagement with the wider public. So we have a Science Cafe every second Monday during the summer month. If you are interested in joining us again, you can go to mdibl.org/events, and it will have a list of our upcoming Science Cafes. And if you're really interested in learning more about what MDIBL does, go to mdibl.org/subscribe, and you can subscribe to our e-news where we share our research as well as other events that we have coming up. Thank you, everybody. It's been really wonderful to have you, and we'll see you all next time.
Thank you. Thank you so much.
Thanks, Anna.